Okay, hello. So I'm going to go through this worksheet now. This one is more complex, going to be honest. So this is definitely a lot of content that we would not have covered in general bio, and linked genes can be tricky to understand. So the first part of the video here, we're not even going to start solving problems. We're going to be just trying to understand how the genes are located on chromosomes, and we're going to just look at that for a little bit first. And then later on, I'll be going through just some sample problems you might see with linked genes. So up until now, we've been looking at genes that are on different chromosomes, even though I haven't really been specific about that. For instance, we have looked at a lot of pea plant traits. So I'll just kind of use that since that's familiar to everyone. So we have done like dihybrid crosses of individuals that might be heterozygous for two traits. So you know that if you have a pea plant that has like heterozygous for tallness, that it would look like that. And then you know that what are the possible gametes or reproductive cells that could be produced from that? Well, you know the FOIL method, first, right, outer, inner, last. So you know that you could get this, you could get this combination as a gamete. So you could get this one, and you could get that, right? So we've already kind of done those types of things. However, what we're going to look at now is I want to actually be more specific and kind of show how that occurs, because I think it's really important to understand what's happening in meiosis here normally, so that we can understand what's happening as well when the genes actually are on the same chromosome. So we'll just go ahead and start out with, a cell. And so this cell, let's say, has the gene for tallness on this chromosome here, and it has a capital T and a lowercase t. So that, because it has two parents. So that would be like chromosome 1 or something like that. And it has two copies, and it's heterozygous. And then let's say that the gene for whether it's round or wrinkled is on another chromosome, totally different chromosome. Right? So this would be like these would be like chromosomes number 1, and this would be like chromosome number 2 or something like that. Okay. I don't even know what number chromosome they're on, but there we go. So this is what this would look like. And it is what it looks like, because the alleles for tall and round or wrinkled are on different chromosomes. So in S phase of interphase, so this is going to duplicate, right? Synthesis phase. And we're going to end up with a cell that has chromosomes that now kind of look like X's, because there's two chromatids attached at a centromere. Okay, and then now you have also duplicated all the genes on each of those chromosomes. So I'm going to show that. And these are the little t and the little r. And the reason I'm kind of showing them like this, let's say those are from biological mother, and let's say that these are from biological father. The reason I'm showing them the way they are, where the biological father basically gave both the dominant traits, and the biological mother gave both the recessive traits, is I'm kind of assuming that the plant that we're looking at was the result of a cross that would have looked like this. So that the little t's and the little r's would have been inherited together, and the big t's and the big r's would have been inherited together. So that's kind of the reason I'm showing that. And this would be like the F1 generation of that parent generation cross. Anyway, okay, so now after S phase of interphase, let's say that this is going to end up becoming a reproductive cell. We have to go through meiosis now. And remember, in meiosis 1, which is the first division, that we have these things lining up. So let's just pretend that this cell that we're looking at actually is in meiosis 1, and that the genes from the father are on the left and the genes from the mother are on the right. And then we're going to go ahead and go through meiosis 1, where these are going to split apart. So So then we're going to end up, this is a good review of meiosis anyway. We're going to end up with that in one of them, and we're going to end up with that in another one. Okay, and then these are going to go through meiosis 2, and then these are going to split again, and we're going to end up with 4 cells, right? 4 reproductive cells called gametes, and these are going to get pulled to opposite sides, and we're going to end up with that. And these are going to pull to opposite sides in meiosis 2, and we're going to end up with that. So there you go. But wait a minute. You're like, might be like, wait a minute, Mr. Buss. All we've shown is that that's possible and that's possible. What happened to those two? Oh yeah, right. So we actually also need to show that there's another way that these could have arranged. So that outcome is possible, but also in meiosis through independent assortment, I'll show over here too. Maybe I'll do this here. That either this or this is going to happen. And what we didn't show was that these chromosomes could have lined up differently, or maybe I'll show it like this. Depending on how they lined up, they could have lined up in metaphase 1, like on the left, or they could have lined up like this. Okay, and then when this thing split, see how we're going to get different combinations there, and then we're going to go through. There's meiosis 1. And then when these things split again in meiosis 2, we're going to end up with these combinations, with these cells. That they'll either be big T, little r, and I'll write that, or they'll be little t, oops, little t, big R. And now you can see, right, that those have occurred as well, and that we have, equal proportions of all 4 of those gametes being produced. Okay, so that's what normally happens. Well, that's what happens in meiosis. And that shows how we get to these 25% chances of those 4 different genotypes if the genes are located on different chromosomes. So I'm actually going to create another video. I'm going to pause it because the videos are going to be longer here. But I think we need to do that because these are difficult concepts here. I'm going to show what happens next in the next video of the playlist, what's going to go on in meiosis if the genes are actually on the same chromosome.